she is a postdoctoral fellow in the center. And I really like that presentation because it will tell you about the actually, where those microfluidics are employed in commercial applications. We talk a lot about the research, right? And of course, that's our life. But those microfluidics are actually, a workhorse of very many commercial applications and sold and applicable in by big vendors, as you'll see. Thank you.
Thank you, Dr. Maggie, for that nice introduction. And yes, we have been listening to some great talks from speakers since yesterday about how microfluidics and nanofluidics are beneficial for different applications and how it has been implemented in devices. And in this talk, I will be giving an overview on microfluidics and nanofluidics, where it stands in this global market, especially in commercial applications. So we can classify this microfluidic and nanofluidic market into three parts. The first one is materials. So materials, yes, we talked about silicon, glass, and polymer. When it comes to polymers, we talked about PDMS, especially about thermoplastics. So yesterday, Dr. Soper gave a nice talk about how it can be used for mass production. And this morning, Dr. Maggie gave a nice presentation on how we can use for surface modification and how those modification can be used to attack for our targets. And many other benefits. And the next one is components. Yes, you just heard a nice talk from Katie, how these nice micropumps, microvalves, and all other microneedles and all those things have been integrated into a fluidic system to manipulate or control all our samples and other reagents. Applications. Now, we can give a handful of examples on this application. Here, I'm giving you a few cartoons. The first one is organ on a chip, which we don't have to actually use a human or an animal subject to do an experiment, but instead of that, you can use cells from this specific organ and immobilize or grow it inside a microfluidic device. And use that microfluidic device to mimic that organ. And the second one is point of care devices, which means you don't have to use sophisticated instruments, but just use that one drop of your sample inside a microfluidic device, and you can get the results intended within probably like few minutes. The last one is for drug discovery, or how efficient the drug will work for a specific disease or for a disease. And as I mentioned, in this one, you can immobilize the cells or grow cells inside a microfluidic device and introduce the drug and then see how efficient that drug is working on that specific disease or for that cells. So in 2018, the global microfluidic market was valued around 5.1 billion, and it's increasing and projected to reach around 75 billion by 2032. And with here, you can see what kind of areas we are using these microfluidic nanofluidic devices: hospital, clinics, homes. So when it comes to home, we know like great examples like COVID-19 at-home test and pregnancy test. So everything it made our life easier because we didn't have to go to a hospital or a clinic to get all these things done. So this graph gives you an idea like how things will be changing and what kind of market that we have for point of care devices, especially in US. And yes, so not even like when we think about point of care devices, even the government is inviting the private investors to invest these kind of projects because we are in higher need of these devices. Even in pharmaceutical industry, you know, when we go through a drug discovery process, it involves several steps. So even if we can miniaturize some of these steps, it will help to reduce the cost of the whole production. And even some of the companies are already using these kind of microfluidic devices in their whole process. So this table gives you a nice idea about what kind of microfluidic products that we have, which are commercially available. And we definitely can add more into this table. And with this, I will start explaining few of the microfluidic devices which are used commercially available. And I will start with my first microfluidic device, and I will explain nanofluidic devices later. So Agilent 2100 bioanalyzer, it's an automated electrophoresis tool which uses a plastic chip. So when you think about normal gel electrophoresis, a slab gel electrophoresis, it takes about 2 to 3 hours for whole sample loading, running the gel, and staining, destaining. So it's time consuming. And it requires higher sample volumes according to the concentration of samples that you have as well. But in here, you can easily use lower sample volumes, about 1 to 5 microliters inside this chip. And it's straightforward. You are applying all the sample, gel, conditioning, destaining reagents, and even the ladders into the same device. And you just keep it inside the instrument. And you can get the results within about 5 to 30 minutes. And it's a disposable chip, and it gives you the flexibility of running it fast as well as without any cross-contamination. The second one is Agilent tape station. We are highly using these screen tapes in our lab. These are made with plastic, and they are injection molded. All the gels, it's an electrophoresis device as well. So all the separating matrices are inside these lanes. So what we do when we introduce these screen tapes into the instrument, and we are placing our samples inside the instrument, so automatically picks the sample and load into the channels, and it gives a nice gel image. Even if you can the instrument itself, it can calculate, it can measure how much of the sample, how much of specific reagent that we had in our sample as well. The third one is next generation sequencing from Illumina. So it's a nice application of billions of nanowires inside these lanes. You can see they are patterned. So why it's important? If it's not patterned well, if it's randomly all over the place, it will not help you to get a nice readout. But when you pattern the cell with these nice nanowells, each nanowell will be only holding one target. So with this pattern flow cell, it will increase your readout, and it will help to improve the quality of your data as well. With that, the next one is microarray hybridization for nucleic acids from Affymetrix. This is an gene expression technique as well. It has the same concept which we saw in the previous example, but in a different way. So in here, we are not using any nanowells or microwells, but we are using a photolithography process for creating DNA microarrays. On a wafer. So what happens is the oligonucleotide probe is located in a specific location where the labeled RNA target will be attaching. So again, you can see how localized they are. If it was random, it will not help to get an idea about the whole sample. But here, when it's localized, you can individually check on each target that it was introduced into each specific location. The next one is droplet generator from Biorad. So why droplet generator is important? If you think about normal PCR, and if you have one sample, when you will be reading a fluorescent readout at the end of the PCR, and it will give you an overall idea about the sample with that fluorescence reading. But if we can make that sample into small droplets, so that's what droplet generator is doing. Literally, you can introduce sample and oil into this plastic device, and it can make up to around 20,000 nanoliter size droplets, and those droplets will be used in 96 well plates, which will be used in the thermocycler to do the further analysis for the digital PCR. The next one is a Nadia instrument for single cell analysis. So it has the same concept where we are using droplets, but in here, not just single DNAs or single RNAs, but the whole cell. So we are introducing cells. The device is manufactured to introduce cells with beads, and then they are encapsulating these combinations of cells and beads into a small oil droplet, which is encapsulated within with an oil layer. And as you can see here, so you can go up like 50,000 single cells can be captured per cartridge. So this is another injection molded plastic chip. The next one is integrated fluidic circuit. So Katie explained a lot about these integrated systems. So this one is actually a hybrid of PDMS and plastic. So the whole white color over here is actually plastic, and then in the middle there is all it has about 9,000 different chambers, like working individually, which is connected with microfluidic channels into these assay and sample regions. So the nice thing about this is it's not just one assay. You can even use more than one assay for the same sample or for more than one samples, and then everything will be controlled using the valves and interconnects here. And Fluidime was the company which was introducing this one, but from 2022, it's called as Stand Biotools. And it was the first company to commercialize digital PCR in 2006, and public offering started from 2011. The next one is a single cell Western blot platform. So again, this gives us high flexibility to work for single cell level. Why? When we do an experiment for a whole sample, we cannot look at each single cell. But in this platform, it gives you the flexibility of looking at each single cell up to about like 1,000 cells per each run. Per cartridge, you can have around 1,000 cells run simultaneously at the same time. So you load the cells, wash, lyse cells, and everything will be happening inside these small chambers you can see here. So all at the same time, it gives you the flexibility of you identifying more than 12 proteins per cell using variety of strategies. Next one is real cell sorting from Depare from Menarini Silicon Biosystems. Dr. Maggie gave a nice talk about how much these real cells are important when we come to different disease monitoring. So as an example, if you think about CTCs, circulating tumor cells, when we have a sample of circulating tumor cells, we have to first label them and then add that sample into this microfluidic device, which is made up with plastic. And each chamber is called DEP cage because we are using dielectrophoretic forces to control where the cell need to go. We can control it using dielectrophoretic forces. So we can control, each cage can be imaged using fluorescence microscopy, and then we can let the instrument to know it should go to this parking area where we can recover the cells from at the end of this whole analysis. The next one is microfluidic blood analyzers from which is iStat system from Abbott. It is another nice point of care device example, which you can use at bedside of the patient. Again, they are made up with plastics, and according to what kind of analysis that you need to do, either it's in endocrinology, blood gases, or hematology, you have to just choose what kind of chip that you want to use, and you will get the specific assays for that as well. You just have to load everything, load the sample, and then it will give a nice readout within few minutes. The next one is veterinary diagnostics from Abaxis. Actually, Abaxis is working in partner with Zoetis. Zoetis is the world's largest manufacturer for medicines and vaccines for the animals. And in this one, even this one, Katie showed, it's a centrifugal system. So you add your samples from here, and then it's rotating inside this in this instrument. And again, depending on what type of profile that you want to know about the animal, you can choose what you want. And mainly the sample can be whole blood, serum, or plasma. The next one is, now we are moving towards nanofluidic devices. This is our favorite for our lab, which is Oxford Nanopore Technologies. It's a nanopore-based sensor. This morning, Hannah gave you an idea about what kind of what are this alpha hemolysin. So it's kind of a pore which all the DNA can go through the pore, and it contains in a membrane which is highly electrically resistant. So when it goes through the pore with the applied voltage, you will be getting a signal, a current signal. Even though I'm just saying just one pore, this whole sensor, it has an array of microscaffolds. Each microscaffold has a pore. So that means we have millions of microscaffolds in this array, and it interconnects together to give you a readout. So Minion was the first handheld nanopore DNA sequencer introduced from Oxford Nanopore Technologies. But and this is the whole flow cell that they are inserting into this Minion, and only in this region which you can see the sensor area. Depending on how much data that you want to collect, they have already introduced a few different flow cells, and Flanger is the lowest readouts that you can get, and with Promethean you can go up like 290 GB. And then Pacific Biosciences, again, it's a nice combination of microfluidics and nanofluidics. In here, they are using glass substrate and about 100 nanometer diameter wells. So what happens is, each well, we are introducing the light from the later part of the from bottom of the well. Because the wavelength of the light is too high, it cannot escape from this nano well, and each nano well we are saying those as this zero mode waveguides as well. So each zero mode waveguide would have one DNA strand attached to a polymerase, and then we are introducing these labeled nucleotides. when the labeled nucleotide is attaching with this polymerase, it will emit a light depending on which nucleotide attached, which can be imaged. So we can analyze all these images and to get an idea what kind of sequencing that we had in the sample. The last but not the least, we are talking about optical genome mapping. This cipher chip from Bionanogenomics. So this chip is basically used for identify chromosomal rearrangements, specifically for some kind of diseases. So what happens is, first we have to isolate the DNAs from the sample and then label them. It will be labeled at specific regions, and then we introduce into the system. In this device, we have like more than 120,000 nanochannels in parallel, and inside the device, all the coiled DNA will be decoiled and stretched. So what they are doing is they are imaging each part of the run, and then at the end, they are analyzing all these images. So that's how this is kind of an example of that. And then, so we talked about different devices from different companies, and actually sometimes those devices are not made inside that same company. Some other companies making that device for that specific company. As an example, herringbone mixer chip. We talked about this specific feature before, which is specifically used for mixing. And actually this chip is made from a Microfluidics chip shop, and not just Microfluidics chip shop. There are some other Microfluidics chip manufacturers in the market right now. These are only a few of them. And as conclusion, we went through a lot of examples from Microfluidics and Nanofluidics, and we can see there's a big disproportion between Microfluidics and Nanofluidic devices. Why? Microfluidic regime is well studied at the moment, but when it comes to nanofluidics, the physics and the chemistry will not act the same. So as a center and as researchers, we are researching on how these differences, or what we know about microfluidics, how we can implement in the nanofluidic area, and what kind of other factors is involved in this nanofluidics. And yes, we are hoping for the best, and we are trying our best. And that's the end of this talk, and thank you for listening.